Dear student, welcome to the basic genetics course and myself Dr. Richa Arya. So today we are discussing our unit 7 week 7 and today's discussion is going to be about mapping of the genes. So I will help you understand how we really map the physical position of the genes on a chromosome, and we will take the example of 2-point and 3-point test process to understand the concept. So first of all, just maybe little basic, maybe some of you are already aware of it, I am just repeating it for the convenience sake so that we are all on the same page. So what exactly the 2-point mean? So 2-point means we are having two genes, right, and we are trying to understand what is their frequency of recombination, right, in what frequency these new combinations are produced or not produced, right. So we are taking two genes in the context, and this is our old example that we have taken earlier too. We have this purple eye and vestigial wing Drosophila, and we have wild type eye and wild type wing Drosophila as parent, and in F1 generation we have heterozygotes, right. And when we cross these heterozygotes with the tester male, it's got shifted here, this is this male, and we analyze the progeny, that is when we start seeing the new combination in the test process. So we have seen this example before, just I am using the same example so that you feel familiar and we can discuss the things in continuation. We did mention that we do see that whatever the parent class is, that dominates because they are the parental genotype, the homozygous wild type and homozygous for purple and vestigial mutation, and the recombinant classes are low. I have discussed in the previous class as well, and these are these recombination classes. We did mention that when we sum the recombinant number, divide by the total number, multiply by 100, we get the recombination frequency. So far we have already discussed these things earlier, just so that put everything in continuation. I am just reminding, now we have this recombination frequency here. So using this recombination frequency, now you see the new use of recombination frequency, we are using this recombination frequency to tell the distance. So what we are trying to say, that we are using this recombination frequency to probably learn about the how far or how close two genes are with each other. So this recombination frequency, the one thing that you need to remember is that what is the relationship between recombination frequency and map unit. You will see map unit many times when people are talking about the distance between the two genes. So the relationship is that recombination frequency, which means 1% recombination, means a value of 0.01 is 1 map unit. So if there is 1% recombination, the distance is 1 map unit, and 10% recombination distance is 10 map unit, and so on. Thus by determining the frequency of recombinants, how many number the recombinants we are getting, we can obtain the map distance between the two genes. So see, one thing that I am telling you, this idea which I have written in these 3 lines here, it took lot of effort to really understand what exactly the recombination frequencies are, why they vary, and how these recombination frequencies can be used to make the better sense of the gene interaction or positioning on the chromosome. I will discuss that historical aspect briefly too. But now let us take another example. Before we were having two genes together, right. Now I am trying to say, when we keep on having more and more and more genes at hand, how we will, and we get the map unit distance, how we are going to arrange them, right. So this slide is going to help you understand how we are placing the genes on the chromosome, and also how we can use these map distance in an additive manner to make these kind of placements. So just see, if the distance between the A and B is 5 map unit, which means that the 5% recombination, and if another gene with A and C, the distance between the two is 3 map unit, right. So A and C has 3 map unit distance, A and B have 5 map unit distance, and if I ask you to arrange the A, B and C. So remember, you do not really know if the C can be here or C could be on the other side of A as well, right. It is just 3 map unit apart. So in that case, you can make one combination like this. You can put A in the center, C on one side of A and B on the other side of A. It is fine because A is 5 map unit apart from B and A is 3 map unit apart from C. You can also arrange these genes in this manner where you put C between A and B and you say that C is 3 map unit apart from A and A is 5 unit apart from B. But this recombination, the one that I have written here, 2 map unit, until and unless you see this relation that how far or close C is with B, you cannot be sure if you should put it in this orientation or that orientation. So I hope you understand what I am trying to say, that when we are trying to create these kind of relative position map for gene, we need more and more relative position of independent gene to fix the position of any gene. So in particular case, out of these two alternatives that we have, the one here and the other here, we can only rule out any of the one when we know what is the distance of B and C. If it is 2 map unit, that is fine, but if it is 8 map unit, then this one is fine, right. So this is the way we arrange the gene in an additive manner on a linear chromosome, and again I will show you that it is something that it is not just something that is obviously discovered. It was lot of discussion and experimental things that people, especially Morgan's lab, has done. What was happening in Morgan's lab, that they were having different kind of mutations, right, and they were trying to understand, as I showed you before with the example of purple and vestigial, they were having many other mutations and they are trying to understand their behavior also, right. And so they have several recombination frequencies, and his whole team, him, Bridges and Stritwan, especially him, there are many people who were working in his lab and working on these ideas and keep on debating, and initially they really have no idea how to understand these recombination frequencies and in relation of these different genes, why they are varying, how to arrange them in certain way that it start making sense. And so Alfred Stritwan, he was just an undergraduate student. In one of the lectures of Morgan, he listened him and he liked his work and he started going to his lab, and he liked the environment of the lab so much that he got involved in the research project. And in his book he has written that they were so puzzled about this data that one night he took the data with him, and at home when he was supposed to do his assignment work, he was looking at this data, and there he thought that these recombination frequencies make more sense when you arrange them on a chromosome in a linear manner, and that is how the first map of the gene was created. So Morgan discovered that chromosome has genes, and how they are arranged on the chromosome was also discovered in his lab. So now coming to the 3-point test cross, we did talk about the 2-point test cross, but I already told you that with 2-point test cross we have limited freedom to understand the relationship between the genes. So we need more and more combinations which are more tedious, and in that way doing a 3-point test cross is an important method to understand and identify the gene position. So I will take more time on this slide so that you can understand everything which is written on the slide. So you see, we are going to do a 3-point test cross, but let us just focus everything in one by one. So you can see the parental genotype is vermilion, which is wild type, which means this eye color is wild type, and here it is vermilion. And CV is cross veinless, which is a phenotype in vein, and here you see the mutation, right. So, these flies have a wing phenotype, and then they also have another wing phenotype which is because of CT gene cut. So, there are 3 different genes, one is vestigial, another is cross veinless, and third one is cut. These are the 3 genes, one is eye related and 2 are wing related. Okay. So, you do not have to go into too much of detail, only remember that when we are putting plus, which means that it is wild type, and we are not putting anything in the superscript, they are recessive traits, and when they are homozygous they will show you the phenotype, right. So, this is the parental genotype, and so the gamete that you will get will be B plus CV and CT, and the other gamete that you will get will be V, CV plus and CT plus, remember that. So, just for the convenience sake I am just trying to remind you here. So, these are, remember, one thing that you need to remember is that these are the parental genotype, these are the parental combination. Just so that if we have to solve these kind of problem, we should know where to look at and how to solve them faster, right. So, V plus CV CT and VCV plus CT is a parental combination, and these are the progeny of test cross, right. So, there are 8 combinations that are possible, right, and I will show you towards the end. I have a table to help you understand how we know how many combinations, if we have if we are doing a 3-point test cross, how many gamete combinations we are going to get, or how many phenotypic combinations we are going to get, or genotypic combinations we are going to get. We have 3 heterozygous pair, 2 heterozygous pair, or 5 or 6 and so on, but I have kept it at the end just for the simplicity. But for now, just remember, when you have the 3 heterozygous pairs, you have the 8 gamete type possible, right. And then you can clearly see that the first two are the parental type, and you can also see that they are the majority of the number, right. So, everything matches, right. What we said, that when the genes are linked, we tend to see more parental type combinations compared to the recombinants, right. Now, we have to make sense of all the others, right. So, what we are trying to look when we were split it into a 2-point test cross. So, what we were doing, we were trying to understand what was the distance of A with B, what was the distance of A with C, right, or maybe if I would have the data, then I would check what is the distance of B with C. Then only I will be able to understand who is sitting where. So, one important question that you need to address next. So, first important thing that you should identify is the parental. And the second thing that you need to identify is which gene is in the middle. Remember, when people are writing it as dot, when they are putting dot between the 2 genes, which means that they are indicating that they do not know the order. They have just written them in this order, but these genes could be arranged in different order. So, your second target is to identify the order, right, and or second or third, we can take them together, the distance between the genes, right. So, these are the 2 things that we are going to do now by looking at this remaining data set, right. So, let us first make sense of these classes. They are the lowest, right, you will need them, right. So, there is one easy way, I will show you in detail later also. These classes, the one that are the lowest in number, they are actually the double crossover event, DCO, double crossover event. We will discuss, do not worry about it, but just remember that the numbers where you see the least offsprings, they are the double crossovers, and they can also help us understand about the location of gene, which gene is going to be in the center, right. So, we will discuss this way of identifying the middle of the gene later. Let us go the longer way first, where we are going to identify the distance between the gene. So, now you see how you will be identifying the distance between the genes. So, if you pay attention to the parental combination and the new combinations that are formed, then it will help you see which two genes are actually recombined. So, now let us, parental they are gone. So, now let us start looking from 3. So, 3, you see, if you just look at V and CV, just focused on V and CV, right. At one time we will look at V and CV in all the combination. So, V and CV is clearly a recombinant for V and CV, because in parental gamete V plus and CV were together, or V CV plus were together, right. So, this is a new combination V CV. So, it is a recombinant. We have written R plus here. Now, you see V plus CV plus, this is also a recombinant, because again in the gametes you do not see this combination. So, we put R here too. Same is true as was the case with 3, same is true with the 5. So, it is a recombinant for V and CV. If you look at 6, then what is the 6 is both are plus. Again this is a recombinant for V and CV. If you look at 7, V CV plus, see V CV plus, it is a parental combination. This combination is already present in parent, and that is why we have not written any recombination here. Remember that. I will show you the little catch here, but for now. So, this is not a recombinant, and you see V plus CV, V plus CV, it is a parental type combination. So, we have not written a recombinant here, right. So, same way as we have calculated the distance between VCV, we will calculate the distance between recombinant between VCT, and then we will look at the distance between CVCT. So, all the 3 possible combination we will find the recombinant. I will just go through once, and this one you can understand yourself. So, let us see, these are the 2 parental types, we do not need to look at them. So, see here. So, what we are looking at VCT. So, if you see this third number VCT, VCT plus. So, VCT plus is present in the parent, that is why it is not a recombinant. V plus CT. So, V plus CT already present in parent, that is why it is not a recombinant. VCT not present in recombinant, V plus CT not present in parent, recombinant. VCT not present in parent, recombinant. V plus CT plus not present in parent, that is why it is recombinant, right. So, that is how we look at different gene set at a time, and then try to understand which ones are recombinant and which ones are not, right. Same you do with CV, CT, just look at CV, not in a parental type, CV and CT, CV and CT, CV and CT, and then identify if they are parents or recombinant, right. So, once you identify the recombinant, what you need to do is just sum them up, divide them by the total number that you have, multiply them by 100, and then you have this recombination frequency for individual pair, VCV 18.5, VCT 13.2, VCT CV 6.4%. And when you have these recombination frequencies, you can arrange them on the linear map, because you already have the 3 distances, there is no confusion. If you put CT, they are here, and this way you have put the CT in the middle, right. So, this as in the initial combination they have placed CV in the middle, but when you identified the map distances, you can clearly see that CT is sitting in the middle, right. So, now you change the position, write it the way you have discovered. So, it is VCT CV and VCT plus CV, that is the right combination, okay. So, now the another important point, if you see that you find this 13.2 and 6.4, and you also found that VCV has a distance of 18.5, means this distance is 18.5, right. But if you sum this up, this one, so this distance is 18.5, and if you sum this up it should be 18.52, but it is coming as 19.6, right. So why we are seeing this kind of discrepancy? Why these two numbers are not matching? It's 19.6 and here it is 18.5. Is there a reason for that? So now coming to the answer of this question, and also the other way, quick way to identify the double crossovers. So what actually happens, that we are expecting that one gene will be having crossing over only once, right. So that is fine when genes are close to each other, but when genes are far apart, more distant than each other, then they have more chances to undergo crossing over, right. So now see this particular very peculiar case where a double crossover is shown. So what actually had happened? You see, it was vermilion, right, that was fine, and it got crossover with cut. You already have placed cut in the center, so you got vermilion and cut, fine, and then it again got a crossover, and that is when you got this wild type for cross veinless, which was the parental chromosome, right. So because of these two double crossover event, actually the crossing over took place, but you thought that it is not a recombinant for V and CT, or V plus and CT plus. See, V plus and CT plus, you thought that it is not a, so it is not V plus CT plus, but V plus CV plus, right, here. CT is in the center. So you thought that this is a parental combination, but you get it back because there was a double crossover event. So to get this error corrected, because you have not counted them as a recombinant, ideally there were two recombinants here. So if you add these two R here for CV as well, so you consider that V and CV also undergo recombination here, then if you add these two R here, then your number will be corrected, right. So this is something which we will discuss in more detail in our next class, where we will talk about the interference phenomena. So what is happening when two crossover take place in the vicinity? They interfere with each other, and because of that we get lesser number of recombinant than we are expecting, right. And that is what I was saying, this is a good way and a quick way to identify the middle gene. If in your question you can see that some classes have very less number of recombinant, there are high chances that those are the double crossover. And in double crossover, what you need to match, you only need to match that which gene is changing from the parental combination. So that by understanding that, if you see the parental combination is VCV, is just take this example, VCV plus CT. So which gene changed? This one. Which gene changed? This one, right. And here if you see, this is a parental combination V plus CV, then which gene is changing? This one. So what we need to do is we need to put this in center. Because of this in center, we are getting the parental phenotype back, and these are our double crossover. And so if you do a little more practice, it will be easier. I would highly recommend you to do more exercise. There are several questions online available. At least pick a book and try to solve the questions, and if you have problem, just let me know. And this require little bit of practice. So this is a way by understanding the double crossover help us understand the process of crossing over and generation of new recombinations. So, just to summarize, double crossover tend to mask recombinants as I just mentioned before, as in our example 2. So, distant loci usually appear closer than they really are. So, thus the most accurate map distance are those that we establish between closely linked gene. So, remember, if if CV is even further apart, then there will be even more recombinants happening and we have no way to really see them, and then there will be an error. So, we have to, whenever we are checking the distances, we need to have the genes that are closer to each other, right. If we are seeing that they are far apart, then we need to find some other gene which might be closer to the combination. And linkage maps, this is one thing that you need to remember. I also told you very early in the class that linkage maps merely map the loci in relation to one another. This is a relative map, and this is what we are using to construct the chromosomal map, right. So, we do not know where the loci is present on the chromosome, we only know the relative position of them in relation to each other. So, this is one important point. Now, when we are in genomic era, we go and directly sequence the genome, we really know what exactly from which base to which base a gene is present, right. So, this is the simplified rule that I want you to remember, you see. So, if remember, then when you need to see how many heterozygous genes you have in the in your question of independent assortment, and so these are the numbers written: 1 gene, 2 gene, 3 gene, 4 gene. So, number of different type of gametes are 2 to the power n, because there are 2 different types combinations possible. So, 2 to the power n, you will know how many type of gametes. So, see, for 3 genes in our case example we have 8 combination, and if somebody want to know how many genotypes they are going to produce, so genotypes are homozygous, dominant, and then heterozygous and homozygous recessive. So, 3. So, it is a 3 to the power n numbers that we are going to get, and different number of phenotypes we have, mostly 2, if there are 2 genes, then we have 2 phenotype tall or dwarf. So, then 2 to the power n, which is 2, 4, 8, 16 depending on how many heterozygous gene pairs we have. So, with that I will end with this activity question. It is a little lengthy, but if you solve this problem, you will definitely learn how to solve the problem of 3-point test cross, and then it will be easier for you to understand more when we will talk about interference and coincidence in our next class. So, this question is where you have to determine the gene order and the map distance between 3 genes. So, they are heterozygous individuals, right. This is a heterozygous individual which is crossed with a homozygous recessive individual, it is a test cross, and the total number of progeny are 1000. And here they have given you individual combinations, the 8 combinations that are possible, and you need to calculate the gene order. You need to also tell the distance between the gene, and I will say follow this template, the example that I have given you, and I have discussed it in very long detail. Just identify the distance as the distances are written here between A, B and C. That would help you solve this question. So, with that I thank you all for your attention, and same as I say in my every class, if you have any question, please feel free to contact me. Thank you once again.